say almost 79 to 83, 101 discoveries that were reported. Only 5 could get a license for clinical use. That means after phase 3 clinical trials, only out of 101, 5 succeeded, and only 1 was a popular treatment in clinical use. So that shows you an idea that it's not easy to get a new product out of an idea. So this is basically a model what we have adopted, and it's a simplistic way how to go ahead and plan your research idea or research project. So basically here at the forefront is not unlike other you will see other unlike other areas where the pharmaceutics department or the chemical and research and development. Here it is the medical affairs and clinical research is at the center of this. So you need to have a good insight of disease on the one hand, and you need to have also a good insight of what are the products which are used or what are the therapy which is currently available to treat that disease. So once you have an idea of both, insight of both disease as well as the drugs which are used to treat that disease, then you identify therapy gaps. So that's the most crucial step. So you just alone insight of disease is not enough. Just insight of therapy area is not enough. So you need to have both insight of disease and insight of products used to treat that disease. And you identify therapy gaps. That's the crucial step. And then once you identify that therapy, the gap which is there in that therapy, then you build your innovation around that. You have a therapeutic rationale and you develop your hypothesis. Then you explore your hypothesis by doing clinical studies. Then you generate clinical trial data, and that clinical trial data you seek regulatory approval through that data. And also you publish your studies, and you can use that data also for promotion of your idea. So this is the model. And I think that's a very, very important area, how to innovate in the medical affairs, how medical affairs and clinical research can play an important role in innovation. And that's the whole presentation is centered around the role of medical and affairs and clinical research. Now, into the various projects which we will discuss, I would like to just give you a brief idea of various study designs. In short, you can divide into the clinical studies into 2. One is interventional studies, and other are the observational studies. In interventional studies, the intervention of the researcher is there, so that you use an active therapy, active drug, or active device, or an operative procedure, whichever you are using, and then you subject them to clinical trials, which could be non-randomized clinical trials when you don't randomize them statistically, or randomized clinical trials. But randomized clinical trials are considered to double-blind randomized clinical trials are considered to be the gold standard for of evidence, so that this is also much acceptable to the regulatory authorities. Then other is the observational studies, where subjects are just observed and no action is there from the researcher's side. So these observational studies are further important because it is not easy to carry out clinical trials. So observational studies have a very, very important role to play. So this could be prospective observational studies and retrospective observational studies. Then you analyze retrospectively a data of a disease or its risk factor, it becomes a retrospective observation. While when you prospectively want to see a particular outcome, then it becomes a prospective observational studies, and which is obviously more important as compared to retrospective. So now just a brief of these clinical studies. Observational studies are important for hypothesis generation, to uncover the risk factors for a particular disease and the effect of therapy which is observed in observational studies. While interventional studies are there for proof, with them you can have proof of your hypothesis, and RCTs, as I explained earlier, are gold standard of evidence, and they are the basis for regulatory approval. So you will increasingly hear the term called as real world evidence. So real world evidence is now gaining more and more importance, and This is nothing but documentation of actual clinical practice. When the drug is approved, then it is being used in the real world. So doctors are prescribing, and patients are using those drugs. So this is very, very important tool, because this will confirm the actual efficacy of the drug in the clinical practice setting, or safety of the drug in that setting. Because once you in clinical trials, you go by strict inclusion and exclusion criteria. So you exclude many patients, but in clinical practice you don't have strict inclusion and exclusion criteria. So you are exposing them to a wider population of patients, and then you actually, and since it is used in lakhs of patients, so safety issues are further uncovered during this. So now this real world evidence, and as such, as I told you, double blind randomized control trials are not visible very, very widely. So this evidence is now being looked at very seriously by even regulators. So that's the importance of, that's why the real world evidence is assuming importance. Now, what are the initiatives which we have taken at Ipca? I think I will confine myself to 4 ideas which research projects which we have worked. And it is important that one needs to be in touch with what is happening in the literature currently, and then in the area of your interest what is happening, and then based on that serves the background of how to think of something. Think of something different. What is the problem, which is clinical problem, which is there in the current therapy, and how you can innovate around that. So that is an example. So at everywhere you will find that there is a background available, and based on that background, there is a need gap which is there in the therapy, and that you can fill that with your research idea. So these are mainly 4 areas where we have conceived the projects around those ideas. First is the safety of NSAIDs, where people are really concerned about the safety of the currently marketed NSAIDs, and how we took advantage of that to give out a more safer NSAID. Then second one is a drug which is an evidence-based thiazide like diuretic chlorthalidone, which has been out of clinical practice for long, and how we played an important role in bringing this drug to the forefront of hypertension management. And third area is again now for inflammation, in particular the chronic inflammation is considered to play a very important role in causation of and progression of diabetes and its complications. So there is the link between diabetes and inflammation is now very well established. So approval of the first anti-inflammatory drug, hydroxychloroquine, in type 2 diabetes. India is the first country to approve this drug. I will just briefly talk about that. And then the last project I want to discuss is about clopidogrel. Clopidogrel is a very, very widely used antiplatelet agent, and there are some limitations of clopidogrel, and which we will be overcoming with our research project. So these are the 4 areas which I would like to discuss. So first is the therapy gap of anti-inflammatory drugs fulfilled by a drug called as aceclofenac. So the background of this project is in November 2000, Merck Sharp and Dohme, that is in short Merck, which is widely known as, it published the results of a study called as Vigor study, where its product rofecoxib, which is highly selective COX-2 inhibitor, that was compared to naproxen in patients of rheumatoid arthritis. The idea of highly selective COX-2 inhibitors is that they don't inhibit COX-1, which is concerned with gastric mucosal prostaglandin synthesis. So if you don't inhibit COX-1, then you have GI safety. Conventional NSAIDs were known to inhibit both COX-2 and COX-1, and when you inhibit COX-1, you there are more GI side effects. So what the outcome was on the expected lines, the GI events were less, but one unexpected outcome came was that there was higher incidence of myocardial infarction in patients in the rofecoxib arm compared to the naproxen arm, which was the standard control arm. So with this, you can see here that there are more CV events in the rofecoxib arm compared to the naproxen arm. And this actually, when what Merck did was Merck explained to US FDA and all the regulators around the globe that the comparator here is naproxen. Naproxen is aspirin-like, so it is naproxen which is cardioprotective, and we are cardiac neutral. And that explanation was acceptable to all the regulators, including US FDA. But that explanation, we were not very convinced on that explanation. So when you have a non-selective drugs, you end up with excess GI side effects. But when you become highly selective towards COX-2, what you are doing is you are not inhibiting COX-1. So thromboxane generation continues. While since you inhibit COX-2 completely, you are actually ending up with inhibition of prostacyclin synthesis. So that is responsible for more thrombosis, hypertension, and excess CV events. And this was in the research in the post-marketing surveillance studies, which were commissioned by the UK by WHO's Adverse Event Center at Uppsala, which noticed that there were less adverse events with the drug aceclofenac compared to other NSAIDs. And then the European Commission then asked 28 European major universities in different European countries to, collate the data of NSAID users and see the safety of those 13 NSAIDs. And it was found that the adverse events, GI adverse events, were less with aceclofenac compared to the other comparators, most notably the diclofenac. And the congestive heart failure incidence was also found to be reported to be less with this particular drug. Here you can see that case control studies in the four European countries suggest that the congestive heart failure is lesser there. So what we did was we identified the need for, neither, if you are non-selective, you conventional NSAID, you have more GI safety. If you are highly selective, you end up with CV safety at the cost of GI safety. So there is a need to have a balance or preferential COX-2 inhibitor where you inhibit COX-2, but at the same time you don't leave COX-1 unopposed. And with that, we launched aceclofenac with the brand name Zerodol in the Indian market. And what we found that subsequently the selective COX-2 inhibitors like rofecoxib, valdecoxib, they were subsequently banned because of causing more MI and stroke. So every other company in India, followed with us, and they introduced aceclofenac since it's a generic product. So all they introduce it. So this is our paper where this is published in Current Medical Research Occasion, where we compared aceclofenac with the diclofenac. And in a large study involving around 600 patients at 21 academic centers across India, and compared aceclofenac with diclofenac. So what you are seeing here is that at each time point of duration of 6 weeks, you found that aceclofenac has lesser GI side effects compared to diclofenac. Another important point you will notice is that these GI side effects, as you increase the duration, the GI side effects increase. So that's why the NSAIDs are advised to you use it on the need basis, and don't make a prolonged use of the NSAIDs, because with increased duration also the adverse events will increase. So that's another important point to be noted there. Then the innovator had aceclofenac only in twice daily doses. So we looked at the pharmacokinetic profile of aceclofenac, and we conceived controlled release once daily preparation. So we had that once daily preparations, and then we introduced it. And the phase 3 data we was published in the official journal of American Pain Society, the Journal of Pain. Then we also introduced various combinations of aceclofenac, which again were useful, and they are today they are the leading products. We have the aceclofenac plus paracetamol combination compared to aceclofenac alone in patients of osteoarthritis flare-up, and the combination was demonstrated to perform better as compared to aceclofenac monotherapy. Then we also introduced a combination of aceclofenac with tizanidine in patients of acute low back pain, and this study was published in Official Journal of European Spine Society. Subsequently, this study has also been referred to in the guidelines of American College of Physicians for low back pain. So, an Indian study featuring in the guideline of American ACP guideline. Then we also a combination of aceclofenac with dotaverine, and this study was done in patients of dysmenorrhea. Again, this study was published in the Official Journal of European Society of Obstetrics and Gynecology. So this is about the introduction of a safer NSAID, which is aceclofenac, and now currently it is the most it is the number one NSAID selling in India. So that's one idea how to do. Now I would like to go to the second concept, which is how we conceive this idea of focusing on chlorthalidone. So this was again the background of this study comes from the literature. There is the largest hypertension trial, which is done by the National Institute of Health US, and these trials involved almost 42,442 patients, and these patients have been enrolled in the study, and the study carried on for almost 8 years. And here this study was published in December 2002, and the study was at that time the newer drugs were amlodipine, calcium channel blocker, lisinopril, ACE inhibitor, and the alpha blocker doxazocine, and they were compared to chlorthalidone. And at the end of the study period, what came as a surprise to everybody is that while primary endpoint of fatal coronary heart disease, non-fatal MI, chlorthalidone was all agents were similar. But in the secondary endpoints, since it's a huge study, even the secondary endpoints becomes very important. Chlorthalidone was better in preventing congestive heart failure to both amlodipine and lisinopril. So this was, and the combined CVD events, it was better than lisinopril. So the finding is that a drug like 60-year-old product like chlorthalidone was better compared to newer agents. So so this was very interesting. And then we wanted to work on this drug. The drug has been was used as a antihypertensive. I had used as a resident this drug in 1980s, but subsequently the drug just went away from the market. So the literature was suggesting that the chlorthalidone is twice as potent as hydrochlorothiazide. So for 50 milligram hydrochlorothiazide, corresponding dose of 25 milligram was there. For 25 milligram of hydrochlorothiazide, 12.5 chlorthalidone was there. But 12.5 milligram hydrochlorothiazide, there was no corresponding dose available for chlorthalidone. And as you know, that 12.5 milligram hydrochlorothiazide is the commonest drug which is used not only alone but also in combination therapy. Not only in India but even all across the globe, 12.5 milligram hydrochlorothiazide is the commonest thiazide diuretic being used. But corresponding to that, there was no dose available for chlorthalidone. So that's how we conceived the 6.25 milligram dose. The advantage of 6.25 milligram dose is that the diuretic potency is also related to its metabolic and electrolyte disturbances. So if you use a low dose, you get lesser metabolic and electrolyte disturbances. And that's how 12.5 milligram of hydrochlorothiazide is very widely used. And then once you use this and the patient's blood pressure is controlled, whether alone or in combination, then you don't need to escalate. So, but if the blood pressure is not controlled, you always have the option of using the higher dose of 12.5 milligram chlorothiazide. So that's how we conceived this dose of 6 milligram. And we did a series of studies, and we showed that it is alone as well as in combination, chlorothiazide is as good as 6.25 is as good as 12.5 milligram of hydrochlorothiazide. So this is a study where atenolol plus low dose chlorothiazide was shown to be effective. Then we had first time we conceived a combination of an angiotensin receptor blocker losartan with chlorothiazide 6.25, and we showed that it is as good as losartan hydrochlorothiazide 12.5 milligram combination. And this data was then, also appeared, editorial appeared on this trial. Subsequently, we also presented this data at the Jackson Cardiovascular Renal meeting at University of Mississippi, and for this we received a new investigator travel award in US. This is metoprolol succinate extended release with chlorothiazide. A new combination was introduced by us. Then what we did here was this is a study published in 2016, where what we showed was that we compared head-to-head 6.25 chlorothiazide with 12.5 hydrochlorothiazide. But we did it by 24 hours ambulatory blood pressure monitoring. And advantage of chlorothiazide is that it has a very long half-life compared to a short half-life of hydrochlorothiazide. So pharmacokinetically, it has an advantage. So it controls the blood pressure throughout 24 hours, and particularly good action during the night time and early morning hours. Control of blood pressure during night time and early morning hours is important because the CV events take place during that time. So control of blood pressure at that time is important. So this is how this study was. Then this study received publication in Journal of American College of Cardiology. Subsequently, we also this study was referred by the Canada's hypertension guideline 2017 guidelines. So an Indian study again finding being referred to in an international guideline. This is the editorial on our study where the authors commented that time to kick the habit of using hydrochlorothiazide. So very, very strong editorial based on our study appeared in the journal JACC. Also, we were awarded as the Best Clinical and Research Excellence Care Award for this study. And among all the multinationals which were present at Boston, an Indian company received this award. So that's another thing. And this year we were pleasantly surprised to see that Braunwald's heart disease covered our study extensively. The recent edition of 11th edition of Braunwald this year, our study was covered extensively. So that's another thing which happened here. It was referred to in 2 chapters. This is our comment, which there was an article in Journal of American Heart Association recommending, pharmacotherapy for pre-hypertensive patients. So then we wrote that whether it is justified to use pharmacological therapy in all pre-hypertensive patients, because you need to see by lowering blood pressure, you also need to lower the CV events. So that's how it is. If you are not able to lower CV events, then whether it is justified or not, that was our point of view. Then commenting on the ALHET study, where on various drugs which you can see amlodipine and lisinopril, the author noted that those patients who do not respond within the first 3 weeks of therapy, they don't fare well. And patients on chlorothalidone were in the chlorothalidone group, there were maximum responders. It is important that to prevent later on CV events, you need to your patients of hypertension to respond early, and chlorothalidone is the one which will help you in responding the patients early during the treatment. This is an observation now on a study published on hyponatremia in patients on thiazide in the Journal of Clinical Investigation. So we wanted the author to consider the syndrome called as SIADS, syndrome of inappropriate ADH secretion. So hyponatremia could be due to excess antidiuretic hormone secretion when the patient suffers from concomitant infections or stroke. ADH secretion takes place, and that results into dilutional hyponatremia. So you need to distinguish between dilutional hyponatremia to thiazide-induced hyponatremia. This was a letter to the editor by us in response to an article in the journal JACC Heart Failure, where the authors, Messerly, they commented in the algorithm of treating hypertension to prevent congestive heart failure. Calcium channel blockers were placed ahead of chlorothalidone in the algorithm. So we said that if the patient has diabetes and diastolic dysfunction, which is very commonly there in diabetic hypertensives, then if you give amlodipine, then there will be increased incidence of congestive heart failure. So important to use thiazide-like diuretic, instead of or ahead of the amlodipine or CCBs like amlodipine to control blood pressure. So this is now the second initiative which we discussed, and it's now chlorothalidone with as CTD is now very widely used diuretic. It is replaced hydrochlorothiazide in India, and after Ipca launched it, several players have entered and they have introduced this. So this is another how you can revive an evidence-based drug. Renaissance has taken place of chlorothalidone, and it is being, India is the only country which has taken advantage of the ALHAT study and and used an evidence-based diuretic like chlorothalidone. Now the next project which I would like to discuss is the diabetes and inflammation link, and how the anti-inflammatory drug hydroxychloroquine has been conceived and used there. So now the link between chronic inflammation and diabetes, not only diabetes but even the cardiovascular disease, there is a chronic inflammation angle there. And this link is being increasingly recognized, and you can see the Time magazine covering this, that inflammation is the secret killer. These are all the references, and now it is very well accepted that chronic inflammation plays a role in causation of diabetes, leading to insulin resistance and beta cell dysfunction. So this is an article which appeared in JAMA in 2007, where it was a prospective observational study of almost close to 5,000 rheumatoid arthritis patients, which were prospectively observed by investigators from University of Pittsburgh for almost 22 years. And as we know very well that as the age increases, there is more incidence of hypertension, there is more incidence of diabetes. So people above 55 have almost 90% risk of hypertension. People above 60 have substantially raised incidence of type 2 diabetes of more than 40%. But here what the investigator noted after observing these rheumatoid patients, they divided these patients into various groups: those who were prescribed hydroxychloroquine for 4 years or more, those who intermittently took hydroxychloroquine, and those who never used hydroxychloroquine. So what the authors noted was that patients of rheumatoid arthritis who were put on hydroxychloroquine for 4 years or more, they took it, they had a much less incidence of diabetes. So this was a very, very, intriguing and very compelling finding that the drug is, preventing diabetes. That's the very strong observational clue. So based on that, then we explored this further. We could not. It was not feasible for us to, carry out a prevention of diabetes study because it required huge number of patients and long-term follow-up for 5 to 10 years, which was not feasible. So we went into details whether somebody has used it as a treatment for diabetes, and we found two studies which were there, small studies which reported that the drug was effective in patients who were uncontrolled on drugs like metformin, sulfonylurea, and even on insulin. So based on that data, we conceived a phase 3 study in 2008 where patients who were uncontrolled on metformin and sulfonylurea, they received, one arm received hydroxychloroquine 400 milligram, another arm received 5-hydrazone 15 milligram. And at the end of 24 weeks, we saw that there was a good glycemic control achieved in both hydroxychloroquine and in hydroxychloroquine arm as good as 5-hydrazone arm. In addition, there was lowering of total cholesterol and LDL cholesterol, which was found only in hydroxychloroquine arm. This was not surprising because hydroxychloroquine is another drug which is there in the market for almost close to 60 years now. And it is widely used for rheumatoid arthritis and lupus patients. And there is an observation that drug improves glycemic, good glycemic effects, favorable glycemic effects are there in these patients. Also favorable lipid effects are there. And the drug has also been found to have antiplatelet and thrombotic effects. So with that observation, we were not surprised with the lipid lowering in addition to glycemic lowering in this study. Based on our data, phase 3 data, we received approval for hydroxychloroquine as a third step drug in type 2 diabetes. Subsequently, this story was also covered in the international news magazine Scrip Intelligence. So besides, the phase 3 study in the type 2 diabetes, we also wanted to study its effect on in patients with dyslipidemia with LDL cholesterol more than 130, primary dyslipidemia. So what we noticed that here we used 10 milligram atorvastatin in one arm and 10 milligram atorva with 200 milligram hydroxychloroquine or HCQ in another arm. So you can notice here that there is greater fall in lipids in the combination arm compared to the monotherapy atorvastatin arm. So this was unexpected line, as we were aware that at least in rheumatoid and lupus patients there is plenty of evidence that there is a beneficial lipid lowering effect of this drug. But this is the first time in non-rheumatoid and non-lupus. First time this is the first study in this population, and also it is the first study combining atorvastatin statin with hydroxychloroquine. So what next we did was we wanted to see what happens to the glycemic status at the baseline and how the drug what happens to these patients at the end of 24 weeks. So what we noticed was that these dyslipidemic patients, 20% were normoglycemic, but around 40% were pre-diabetic and another 40% were diabetic. So 80% were either pre-diabetic or diabetic, and which is as well known because if you have one metabolic abnormality, there is coexistence of other metabolic abnormality as well. So in the diabetes patients, after the end of 24 weeks, there was 0.3% fall in HbA1c, while 0.3% rise in HbA1c, while in combination arm of atorva 10 plus HCQ 200, there was a fall in the glycemic parameters. So even 200 milligram was having around 0.6% drop. Well, more interesting thing happened in the pre-diabetic patients. There was some marginal rise in HbA1c and marginal fall in HbA1c in the combination arm. And then when we studied these patients further, in the pre-diabetes patients, on 15% patients went into frank type 2 diabetes with HbA1c moving from 6 to 7, while in combination arm, the addition of 200 milligram HCQ prevented this, and only one patient went into frank diabetes. And in patients of diabetes, you can see the glycemic status improved in the combination arm of 200 milligram HCQ plus 10 Natorva, while it worsened in the statin monotherapy. So this was our observation in this study. And then we came across this article in BMJ, where investigators from Japan, they reported, increased incidence of diabetes with statins. And not only the high potency statins, but even low potency statins, which was similar to our observation in our data that just 10 milligram atorvastatin in 24 weeks resulted in that. Am I audible?
Yes, sir, you're audible. We can see your slide also.
Okay, good. So you can see that here the similar observation was there. So we wrote that often people underplay, new onset diabetes with statins. But so we wanted to comment on that new onset diabetes with statins, it is a real risk and it is not so trivial what people consider. So that's to show that, and this was published in the letter to editor. Then not only that, we had more authors coming out with similar finding. This is a authors from an American university, where they what they reported was that in patients of rheumatoid arthritis, those patients who were on statins, they had higher hazard ratio for diabetes, and those who are taking glucocorticoid, they had higher hazard ratio, while those who were on hydroxychloroquine, they had less chance of going into diabetes. But this is with the surprising part here is that the hazard ratio for going into diabetes is higher with statins compared to glucocorticoids. So this is one, because normally we associate glucocorticoids or steroids with higher being the most potent diabetogenic agent, but here statin is the hazard ratio is higher. So this was one new thing which we felt was there. Another thing is that while he has reported with individual drugs, the author did not comment what happened when HCQ, in those patients where statin plus HCQ were given together or steroid plus HCQ were given together, what was happening there. So we wrote the author this part and asked him to clarify on these grounds. So the author then came out with the data, and then they explained that those patients who were on concomitant HCQ with steroids and statins, the addition of HCQ was able to lower the hazard ratio for both steroids and statins. So this is another important finding. With steroids, it is known that hydroxychloroquine reduces steroid-induced dysglycemia, steroid-induced dyslipidemia. But here again, the same finding with statins also is noticed. So that's the important thing. Then one more study, which is a landmark study, diabetes prevention program, where the investigators used a pre-diabetic population. They used lifestyle as in the one arm, metformin in another arm, and placebo in another arm as a control. What they noticed was that lifestyle was more effective than metformin in preventing pre-diabetes patient to going into diabetes. But this is a sub-study of that diabetes prevention program. Often people are not aware of this study, neither people discuss this study anywhere. But what was noticed in this study was that almost 1/3 patients of pre-diabetic patients were on statins in this study. So what happened to these patients who were on statins, and in the three groups, what happened? So when these patients were on statin, then all three arms, there was no reduction in diabetes risk. So metformin was ineffective in preventing these patients from going pre-diabetes to diabetes, and even lifestyle was ineffective, which is a worrying part here. So that's an important observation, that both metformin and lifestyle were not able to prevent these patients from going into diabetes. So that's then the third study, since it has been also been shown in the literature that the drug has antiplatelet and antithrombotic effects. We could not find a single study, human study particularly. Animal studies were there, but there was no human study demonstrating its antiplatelet effect. So here uh, we approached the PGI Chandigarh, where it was given as a postgraduate thesis to an DM student, and where the antiplatelet effect of HCQ was shown in healthy human volunteers. This is a proof of concept study, and in combination with aspirin, you can see there is a still better antiplatelet effect. This study has been published in a good journal, Journal of Cardiovascular Pharmacology and Therapy, and a very widely quoted paper now, because that's only evidence for antiplatelet, human evidence for antiplatelet effect. So this is the third study which we carried out. I would like to just briefly mention that now with the approval of any new anti-diabetic agent, the regulators, USFDA has particularly mandated that you need to carry out a CV safety study. Here, we would like to say that now the University of Helsinki has conceived a project where in post-MI patients there will be two arms. One will be a standard arm, and another arm would be, in addition, there would be the hydroxychloroquine would be received, and see the events of CV events after that. So indirectly, a safety study, CV safety study will be carried out in these patients, and this study is called as Oxy trial. The solution for European Heart Journal, but the investigator did not include congestive heart failure as outcome. So we wrote a letter to the investigators to include congestive heart failure as an outcome, both echocardiographic outcomes and clinical outcome, hospitalization for heart failure, and the authors agreed and they modified the protocol and included this in their study. This is a first study of an anti-inflammatory drug being used in patients of acute coronary syndrome, and the first proof of concept that when if you use an anti-inflammatory agent like canakinumab, the CV events go down 14%, 15% risk reduction in CV events. You can see those who were given the anti-inflammatory drug canakinumab versus those who did not receive this drug. So that's the landmark study in that sense, that first proof of concept that if you reduce inflammation, you reduce CV events. But what was important was another endpoint in this study was reduction of patients from pre-diabetes to go into diabetes, but that did not happen. And while we consider, as I explained, that both diabetes and cardiovascular disease have an inflammatory component, here the diabetes was not much affected. There was no prevention of diabetes in patients who were pre-diabetic. So we wrote this letter to the editor where we said that there are multiple inflammatory pathways, and we need to understand that for both CVD and diabetes. So, which, if you have a broader agent, you are more likely to succeed, like hydroxychloroquine, in reducing these CV outcomes. Then, hydroxychloroquine approval has also been, recognized by the RSSDI, or Research Society for Study of Diabetes, official society in India for diabetes, and has appeared there, mentioned there. The ICMR guideline for management of type 2 diabetes also mentioned hydroxychloroquine approval in type 2 diabetes in India. This is an Asian Indian phenotype. The Indians are different than other Indians, are more prone to, often you have heard that India is a diabetes capital, but the reason for that, one of the reasons that our genetic and environmental mix up in us, it makes us more prone to both type 2 diabetes as well as heart disease. And one of the major reason for that is that we have more abdominal fat. Overall, we may not be more obese than the Europeans or Caucasians, but our obesity is more confined to the tummy, and this visceral obesity leads to release of inflammatory cytokines, and this leads to beta cell dysfunction and also to insulin resistance. So we are more insulin resistant as compared to other populations, and we have low levels of adiponectin, which adiponectin is the one which is a beneficial adipokine, and we have a characteristic dyslipidemia, more small dense LDL, reduced HDL, high triglycerides. So this is our Asian Indian phenotype being slightly different than others, and on all on four major factors you can the studies have shown that hydroxychloroquine has favorable effect on these pathophysiological defects. So subsequently, we presented our finding of phase 4 study at the meeting ACE, American Association of Clinical Endocrinology, last year in 2018. This year, we also presented an updated data at American Diabetes Association meeting. Then these are some of the which we generated some intellectual property. So not only you can get the conceive a research project, do the clinical studies, get approval, market the product, but you can also generate intellectual property data around your idea. If the idea is novel, nobody else has thought of before, you can always generate IPR on your idea. So this is best formulation patent award surrounding hydroxychloroquine type 2 diabetes receiving given by Acma. These are some of the intellectual properties generated of hydroxychloroquine in combination with anti-diabetic agents. The US patent for the same. Then combination of hydroxychloroquine with the gliptins, which is patent regarding that. Then combination with statin, patent on that. Then statin-induced diabetes, which observation we made in our clinical trial. So before publishing the trial, what we did was we first applied for patent. So that's an important lesson, that if you have anything novel, first you don't publish it. First go for the patenting if you feel it is worthwhile, and then go for publications. That's what we did here. Because earlier 6.25 we did not patent because we didn't have the idea, but here of chlorothalidone, but here, so we had that lesson, so we published it. We didn't publish the study. First we got the intellectual property. NFLD and NASH studies carried out, and then showing positive effects of hydroxychloroquine, almost reversal of the fatty liver with the combination of HCQ, metformin plus atorvastatin together. Currently, there is no approved therapy available for NFLD NASH. So patent on that in the US granted. Then hydroxychloroquine, there is a lot of data that when you use it in rheumatoid and lupus patients, they don't develop diabetes easily. They have less nephropathy. Lupus patients have less nephropathy. So with that same idea, we also wanted to see it in diabetic nephropathy. So we preclinical models, we showed that it is better effect on diabetic nephropathy. It reduces, slows down this. And this study again, we applied for the patent for diabetic nephropathy. So this is the publication in WIPO. So this was the third initiative. Now coming to the last initiative, where a pharmacogenetic limitation of clopidogrel we are trying to tackle there with a clopidogrel derivative. So there is a box. So as I said, there is a background available in the literature, and this And that with that background, you have to, what is the problem area? You need to understand that problem area and then see whether you can address it. So here, if you read the clopidogrel prescribing information, there is a box warning that patients who lack the CYP cytochrome P450 enzymes, which liver metabolizing enzymes, which metabolize most drugs. Here, the CYP2C19 is, and there is a population which is deficient in this CYP2C19. So you, as per the US FDA prescribing information, you need to see that the patient is not deficient, and then if the patient is deficient, don't use the drug and use alternative agent. So that's the limitation of this drug. So we conceived an innovation to overcome this. So almost around the data suggests, the ICMR data suggests that around 35% of the Indian population either lack this enzyme or they have, this enzyme is less active. So clopidogrel still continues to be widely used in acute coronary syndrome. And this, when you use in patients who lack this enzyme, then you have, diminished platelet inhibition, and then you will have higher incidence of stent thrombosis also. So newer agents are more costly, like prasugrel and ticagrelor, and they are associated with increased risk of bleeding. So that's their limitation in ACS. Further, they are not indicated for, they are only indicated in ACS, but they are not indicated for prophylaxis of stroke and peripheral artery disease. And clopidogrel, while is approved for not only CVS prophylaxis, but also CNS and peripheral artery disease. So that's the advantage. Clopidogrel remains the most widely used antiplatelet drug. So what we have done is we have developed a derivative where it will, convert instead of two step, it will be converted into one single step into its active drug, and this also leads to lesser chances of drug interactions like proton pump inhibitors. So this is what is expected. We have the proof of concept studies in the US population at least, and now we want to study it further in India, where we have applied to DCGI and received, and we have just in principle approval for carrying out phase 1 PKPD study. And some advantages we perceive, and we need to prove that in further studies. So with that, I would conclude that this will give you an idea how to conceive a new idea, and you need not be a chemistry expert and do conceive a new chemical entity, because existing drugs, there is a lot of space for innovation in existing drugs. And the model of multinationals, they can't innovate in this space of the drugs. When it becomes off patent, the multinationals are less likely to innovate in that space, because that model is not rewarding to them. But for generic companies and for all other stakeholders, other than that, this model is good, because the drug for good effects of the drug, the bad effects of the drug, the clinicians are aware, and plus the drug comes at a much economic price. So it is good for everyone, even for the providers it is good. So with that, I would like to end my presentation, and I would like to acknowledge the contribution for our team, clinical research team, and the support from Ipca management for carrying out this research work. And I would like to acknowledge that this drug would, from to my teachers at Tokyo National Medical and Nair Hospital, Mumbai. And this is where I would like to end. I think the time is, I think, probably almost coming close to that. I don't know whether any question answers have been planned, or whether we can we'll wait for further from Sakaram to guide us further. Dr. Sakaram.
Thank you, sir. It was just I know it has been raining heavily in Mumbai since morning. Let me just acknowledge, sir, first. I think I have been meeting you, I have seen you, and the kind of genuine research work. We are physicians, so I know, sir, in the other Indian companies or MNCs, what kind of work happens in India, and the kind of innovative research you had been doing at Ipca, and the way you have transformed a couple of therapeutic areas and developed some unique molecules. I think it's a commendable work, sir. And on this platform, I absolutely appreciate that.
Thank you.
What I suggest is that I can only take one question if somebody has, or I think you can write to me if you have any questions, and I will ensure that I reply back to you. I will not hold back, sir, and Vivek and the other people who are on the call who are in office, because it's pretty late, sir. I was in Andheri and it's raining heavily.
Okay.
So I would just thank you once again, and thanks all the participants who are in the call. Just to make a note that we conduct these webinars every 3 weeks, and I would look forward to, sir, again, maybe next 3 months time or 4 months time, to come back on the same platform. This is our third webinar as such. We have tried to cover the opportunities in medical affairs, medical device industry, and today the innovations in clinical research. So, sir, I will come back to you again in 3 months time with a newer topic, and we would love to hear it from you. Thank you once again, sir, to you and Vivek.
Thanks a lot, Dr. Sakar.
Thanks, Doctor.
Okay, thanks everyone. Thanks for the participation. Thank you, and a nice evening to all of you. Okay, thank you.
Yeah, thanks. स्टॉप कर देते हैं. कितने लोग हैं? हाँ, अभी तो आई थिंक यहाँ पे 12 दिख रहा है मेरे को. कुछ छोड़ दिए होंगे यहाँ पे, तो अभी भी जो है वो आ रहे हैं, पर वो ज्वाइन होते हैं उनके आई थिंक वो नहीं पता चला होंगे, बट वो ठीक है. अभी तो निकल गए. ये स्टॉप कर देता हूँ.